So I'll start here with pyruvate dehydrogenase. Again, three important things you want to know about that. First, where it takes place, mitochondrial matrix only. Okay. Second, what is its function? Metabolic link between glycolysis and the TCA cycle. Third, it will produce energy in the form of NADH. So those three things are very important to start with, as that really covers two things: what is it for and where it takes place. Now, this is a pretty complicated enzyme. Sometimes, or oftentimes, this pathway is referred to as pyruvate dehydrogenase or pyruvate dehydrogenase complex, because in essence it is a complex of multiple subunits, many proteins, as well as coenzymes. So let's get started with this complex. So the terminology, again, if you hear pyruvate dehydrogenase or pyruvate dehydrogenase complex, treat them the same way. They are synonymous. Okay, so we use the two terms, pyruvate dehydrogenase or pyruvate dehydrogenase complex interchangeably. Okay, so this complex contains 5 enzymes and 5 coenzymes. Okay? Easy to remember. 5 and 5. 3 of the enzymes are the catalytic enzymes. Here, pyruvate dehydrogenase, dihydrolipoyl dehydrogenase, and dihydrolipoyl acetylase. These are, all 3 of these are involved in the catalysis, converting pyruvate to acetyl-CoA. Those 3. For that, they need 5 coenzymes. Remember, coenzymes are a type of prosthetic group that are organic in nature. CoA, coenzyme A, F.A.D., N.A.D., thiamine, and lipoic acid. 1, 2, 3, 4, 5. All of those, the 5 coenzymes are needed for catalysis. If one is missing, this thing shuts down. The entire complex shuts down. 4 of these guys are derived from vitamins. That's how most coenzymes are, what most coenzymes are. They're derived from vitamins. So that means we need CoA, F.A.D., N.A.D., and thiamine from the diet. These are provided by the diet. Lipoic acid is in a different color because we can make it. It's not a vitamin, it's a coenzyme, but we can make it. So we can make enough, we don't need to get it from the diet. So I said 5 and 5. There are 2 other enzymes in there, but they play a regulatory function. One of them is called PDH kinase. It's a type of protein kinase. And remember, protein kinases, their function, like protein kinase A, similar to that, is to add a phosphate or phosphates to an enzyme or a protein at specific serine, threonine residues, usually, changing their activity. So, pyruvate dehydrogenase complex contains a PDH kinase. That means that protein kinase can phosphorylate the catalytic enzymes and change their activity. It also contains a protein phosphatase, or also called PDH phosphatase. And the function of that is the reverse of the kinase. It can remove phosphates from serine and threonine residues on the catalytic enzymes and activate the entire complex. Okay? We'll get to that in some detail in a minute, but you need to know that the 5 protein or enzyme components and the 5 coenzyme components of pyruvate dehydrogenase complex. I'm just going through some of these here. Not you need to know anything about structural, really, components, but coenzyme A is one of the coenzymes. CoA is the abbreviation. It's derived from vitamin B5, and you can see it has a nucleotide component. But this is really the important part of the molecule. This is the entire coenzyme A and all of that, but it attaches there. See the sulfhydryl group? That's where it attaches to, say, in this case, acetyl-CoA, and create this high energy bond. Okay? That thioester bond is a high energy bond. Again, don't worry about the details, the structural details. I just want you to have an idea what they look like. These are large molecules, organic molecules derived from vitamins. Here's a simpler one. Here's the acetyl group, and that's the entire CoA molecule is the rest. NAD derived from vitamin B3, again, nucleotide component, nicotinamide adenine dinucleotide. Again, don't worry about the structures, just have an idea what it looks like. It has a nucleotide component. It's derived from vitamin B3. And FAD, similarly, is derived from vitamin B2. Again, it can be reduced to form FADH2, or similarly NAD can be reduced to form NADH. Okay, so I'm going to skip the slide. It's a little too wordy. I'll go to this one, which is a little simpler. So to start with, I want you to know what the reaction is. If you don't know what the reaction is, there's no point in even learning the regulation and all the details. That's very crucial. This is the reaction. Focus on the red molecules. Pyruvate losing a CO2, forming an acetyl-CoA. This is 2 carbons, 1 less carbon, that's the CO2 lost, and attached to CoA. That's the product, acetyl-CoA. And this is again crucial for this and for the TCA cycle. When you lose a carbon, that's typically considered an oxidation. You shorten something. It was 3 carbons, now it's 2 carbons. That's an oxidation reaction. When you oxidize something, going from pyruvate to acetyl-CoA, you have the electrons that were lost, if you want, have to be gained by something else. That's why oxidation and reduction always go hand in hand. Something loses electrons, another thing gains those electrons. Okay? They don't go disappear in thin air. So once you oxidize pyruvate to acetyl-CoA, something else has to be reduced. And the something else that gets reduced is NAD plus, one of the coenzymes, to form NADH. So NADH is electron-rich, high-energy molecules. Anytime you see, forget about the H plus here. That's just to balance the equation. But NADH itself is a high-energy molecule, electron-rich. So every time you oxidize pyruvate by pyruvate dehydrogenase, you produce one acetyl-CoA, one NADH. Here are the important enzymes, the three catalytic enzymes: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. What I want, you don't need to know the exact details here, just to simplify life. You just need to know here are these coenzymes: thiamine pyrophosphate, lipoate, CoA, F.A.D, and N.A.D. Their function is one of two things: either to carry the acetyl group, in the case of thiamine pyrophosphate and lipoate and CoA, they're carrying the acetyl group. Sort of accepting, donating the acetyl group. The other two coenzymes are accepting the electrons. So the first acceptor of the electrons is F.A.D, then convert to F.A.D.H2, and then F.A.D.H2 donates its electron to N.A.D to form N.A.D.H. So just appreciate the role of those coenzymes. And some of them just carry the acetyl group over to the next acceptor, and the others carry the electrons. The key thing, remember what it produces, where this happens, and don't worry about the details of the sequence, okay? But you need to know that these three catalytic enzymes are involved. These are directly involved in the conversion of pyruvate to acetyl-CoA. So pyruvate dehydrogenase complex, it still has, you remember, two other enzymes: the kinase and the phosphatase. The phosphatase function is to dephosphorylate the complex, dephosphorylate the catalytic enzymes, and therefore activate them. So pyruvate dehydrogenase complex is always active in the dephospho state. And what promotes the dephosphorylation is the PDH phosphatase, or protein phosphatase, within that complex. Don't worry about these details here. We're not covering that, but this is kind of important in the big picture. But the key here to remember is dephosphorylation of the complex activates the catalytic enzymes. The reverse is true. Phosphorylation of the catalytic complex inhibits the complex. Phosphorylation of the catalytic enzymes inhibits the enzymes, and you cannot break down pyruvate under those conditions. And this is really summarized here. Here's the pyruvate dehydrogenase. That's the enzyme. Okay? Think of this as the containing the three catalytic subunits. It's active when it is in the dephospho state. That's when it's able to convert pyruvate to acetyl-CoA and producing NADH. When the protein kinase, or again, could be called PDH kinase because it's specific for PDH, is active, it's going to add phosphates, multiple phosphates really, to serine and threonine residues on the enzymes, part of the enzyme complex. And see what happens. They become inactive. What activates this kinase are the two of the end products, acetyl-CoA and NADH. Acetyl-CoA and NADH indirectly activate, they first activate the kinase and convert the complex from the active form. The kinase will convert the complex from the active form to the inactive form. To go back to the active form, typically you need calcium that will activate the phosphoprotein phosphatase. Okay? This one here is a little simpler. I kind of like this one. So here's the active PDH. Again, that's the 3 catalytic enzymes. It's active when there are no phosphorylation on the catalytic enzymes, converting pyruvate to acetyl-CoA plus NADH plus CO2. As these guys accumulate, think about it. When these guys accumulate, let's say pyruvate dehydrogenase is going, you have a lot of acetyl-CoA, a lot of NADH. As these guys accumulate, they will activate allosterically PDH kinase. That's part of the complex. And it will start to add phosphates from ATP to the catalytic enzymes. Here are these phosphates on serine and threonine residues, and the enzyme complex becomes inactive. You can go back to the active form by activating the phosphatase by either calcium or magnesium. Okay? So that's really a crucial slide to understand in terms of the regulation. I just want you to appreciate that what we're getting here, let's say we started there. I want you to just tune into the big picture all the time. Glucose going to pyruvate. We just oxidized pyruvate to acetyl-CoA. If you want to be accurate, this should be 2 pyruvates, and it will give you 2 acetyl-CoAs. Okay? Because glucose cannot give you 1 pyruvate. It gives you always 2, and the 2 pyruvates will be converted. But I want you to appreciate that that acetyl-CoA comes from other pathways as well. Yeah, I want you to appreciate that that acetyl-CoA comes from other pathways as well. And it can come from amino acids. It can come from catabolic processes involving fatty acid beta oxidation. So it's a very important kind of common intermediate in metabolism, not just coming from pyruvate and glucose, but coming from other nutrients that we take in, fats and